My name is Johannes Schmid and I would like to show you how NetworkAnalyst works. This is a free web-based platform for the analysis of differentially expressed genes or proteins following a network analysis on different levels and the comparison with common databases such as Reactome or KEGG. So let us first start with the input of genes. So you have three different possibilities. You can select the list of genes or proteins where the analysis of differential expression has been done already before, for instance with gene expression omnibus and GU2R. Or you can upload your own raw data and let the web platform analyze the differential expression, or you can even upload multiple gene expression data for a meta-analysis. So let's get started with a list of genes or proteins where the analysis has been done already before with respect to differential expression. So when we click here, we enter the next menu. Here we have to define first which organism we analyze, so from which organism the genes come from. So I select here human, and then you have to define what the gene identity type is. So in this case I use official gene symbol, and then I can actually upload the data. I switch now to Excel to show you a list of the genes that I'm interested here. They are now already in this format of gene symbol in comparison to the log fold of control. So I can select here and for instance all the genes up to a certain p-value. For simplicity I just use a restricted setup here. So I copy these lists, then I switch back to the network analyst and I paste the genes into this window area, and then I simply click upload. There you see here then on the upper right side that it actually replaced 8 duplicates. All the IDs are unique. When you click here you see an overview of the data that you uploaded. So it's a number of 70 genes, and now we can actually proceed by clicking here on the proceed button. And then you enter actually already one of the core windows of Network Analyst, which is the Visual Analytics window. Here you have a number of possibilities. So you can choose here protein-protein interactions, gene microRNA interactions, transcription factor gene interactions, or even protein drug or protein chemical interactions. If you uploaded a gene list already with pre-calculated fold of control values, heat map clustering and PCA and so on is not available. I will show you that later. So we start out with protein-protein interactions. When you click here, you will see here there are 3 databases from which you can choose. The interactome, so that is the IMEX interactome, literature curated from a publication as defined here. Then you have the string interactome. Here you can actually define a confidence cutoff, and you can activate here whether you would like to have experimental evidence as a requirement. The Roland interactome is only available for human data, not for mouse data. Actually, in some cases it might make sense to use the human interactome even in case your sample comes from a transgene mouse study, because the human interactome is richer and you will get probably better p-values and a better analysis by comparing the mouse outcome with the human interactome. So, but now let's stick to the IMEX interactome first. Here we click on OK, confirm that, and then you will see that so it tells you that actually everything is okay, and you will see here a list of different subnetworks, and you see that usually the first subnetwork is a very rich one with many nodes and edges, And this is like the continent, the biggest subnetwork, and the other subnetworks are usually kind of islands, smaller islands that might be of lower importance. By default, the system calculates the first order network, but if you have too many nodes, more than 2000 nodes, it might be advisable to choose the zero order network, which will restrict then the number of nodes. If you have not enough nodes, you can actually choose the second order network, which will increase the complexity level. Another quite interesting option is to choose for minimum network, because this is actually the network of the interactome that you chose, that is minimally required to link all the differentially expressed genes that you have in your data set. So, but now I think that the first order network actually makes good sense. So we click here on the proceed button, and then we will see a network analysis window. So it takes some time to calculate it. So first it comes up in this view, and you see here on the upper left that this is subnetwork 1. So you could also go for subnetwork 2. So you see subnetwork 2 is just a rather small island, whereas subnetwork 1 is quite rich. And it gives you first a default layout, but you can change that. And what I often like to do is to click here on a white background, then to change the view from topology to expression, meaning that all the upregulated nodes will be shown in red and all the downregulated nodes in green. And apparently there are more red and green here. And you can change here the layout further to make it better visible. You can try different possibilities here. One that I like quite a lot is Force Atlas, because there it rearranges the network in the way that basically the important nodes are all better visible. So you see that your seeds that you had in your differential gene expression list are listed here rather on the periphery of this network, and all the interacting proteins that had been identified by comparison with the protein interaction network are shown here in gray nodes. You can then also download that if you like in different formats, such as PNG or SVG or GraphML. You can also change here, for instance, the node size. So you can increase here the node size. You can change the edges. You can change how the highlights are shown. Then another thing that you can do here is you can go here for reduce overlap, and then it will calculate an illustration where, for example, where the overlap of the nodes is reduced, and you can then usually read better the labelings. Not all the labels are shown here, but you can change that with more options by playing around with that, so that you can also see, for instance, these other nodes here in between. On the left side of this illustration, you see basically the so-called node explorer. So that gives you some parameters of the network, so the degrees a certain node has, so which nodes, and the betweenness, and so on, and the expression level here. You can save that list here, so it would then open a dialog telling you how you can actually save this list in a CSV format. And when you click on something like here, you will see on the lower left side the current selection, and here you also see then the link to a database entry. So here you could actually just click on that, and it will tell you some information on that node in UniProt in this case. Okay, now we come to the beauty of the whole system, that is the function explorer here on the right side. You can actually choose here between different databases that you would like to query with your subnetwork that you have here. and you can either query all the nodes, or you can actually restrict that to only the upregulated nodes or the downregulated nodes. Basically, CAC is usually not as rich in information, and the statistics is not as good as compared to Reactome. So let's start out with Reactome and all nodes. When we click here on the submit button, we see here these are all the enriched functions of the Reactome database that you find in your interactome of your seeds when compared to the protein interactome. So when we click here on, so apparently messenger RNA splicing is something that pops up here. So when we click on that, we will see then all the nodes here highlighted that have something to do with messenger RNA splicing. You can change the color here, or you could actually now, there's another thing that might be interested, like immune system, and here we can, for instance, change here the color from blue to black. And now when we click here on immune system, you will see all the nodes that have something to do with the immune system will have a black border, and all the nodes that are somehow related to messenger RNA splicing will have a blue border. You can also save this list here again in a CSV format for later analysis, and then there are some other options that you can choose. So I deactivate here. Another interesting function is here the so-called Model Explorer. Models are subnetworks that are much more linked, where the nodes are much more linked as what would be randomly expected. So when we click here, you have 3 different algorithms available. When we click on submit, it will calculate different models. So in this case, we find, for instance, a model here of 62 nodes, but actually just 2 of your seed proteins of your original differentially expressed genes are part of this model. Again, you can change here the color. For instance, here we now go to blue, and then we choose this other model, and the other model would be, for instance, here. So this model would have a size of 9 nodes, and 4 of them come from your differentially expressed genes. With that information, you can actually even extract the nodes by clicking here on that button, extract current highlighted models, and it asks for confirmation. And when you do that, it will calculate a new network, and this new network will be shown up here. That is model 1. So we switch back to the subnetwork 1. Here it automatically goes back to the original illustration, to the original layout, so we can go back to Force Atlas if we would like to get back to this view where basically all the seed proteins are in the periphery of the network. So we can then basically, so I go back to Function Explorer, and we can now, first we did it with all nodes, now we go to upregulated nodes and Reactome and click submit, and now you see if we just look for the upregulated nodes, the interferon signaling would be one of the highest hits. We see here the p-value, so we could actually now again activate, let's make it black here, the interferon signaling, and we could choose several of these pathways or enriched functions like cytokine signaling, and you see that the nodes will get bigger and bigger when we do that. So when we highlight more of these Reactome pathways, we will see here then all the members of these pathways. The nodes will become bigger, and also the labeling will show up for the different nodes. Okay, now we could actually go to biological process, and we click submit, And then you will see here response to virus would be one of the biological processes, but we still keep the layout from before. So these nodes that we had selected with reactome comparison are still labeled here in black. And now, for example, we could switch to another labeling color and click here response to virus. Then we'll see that some of these will be the same and others probably are not overlapping. But here we could then again activate here different pathways that have a low p-value, so that are significantly enriched here among our differentially expressed genes. And in this way we can actually go through all these different possibilities. If we want to switch back to the original view without the selection of these nodes, we can go always here to reset to default view, so then this labeling is gone again. Here we can actually do an auto fit of the network. We can zoom in, we can zoom out, we can move up and down and left and right, and this here is to manually select and drag multiple nodes if necessary. So now we look what happens if we query only the upregulated nodes against the database of molecular functions. So here you will see, which has again something to do with viral defense. We see double-stranded RNA binding and so on. And now, for example, we try gene ontology, cellular compartments, and there we will see that the upregulated nodes belong mostly to the cytosol, but some also belong to the extracellular space or cell surface. And actually analyzing the data set with these different comparisons gives you already quite a good insight into which direction your differentially expressed genes actually go, or what kind of change they are doing to the cell or the organism that you were investigating. So finally we can check for motif. This gives you the transcription factor binding motif. So when we click submit here, we will see here that serum response elements are increased with that low p-value, and interferon response factors and so on. So we see here IRF7, IRF1 and so on. So we see the different transcription factor binding motifs that are upregulated among our genes. And then of course you can do the same with downregulated nodes, which might, so let's just go through. So that is the downregulated nodes would indicate leukocyte transendothelial migration, tight junction, cell adhesion molecules. That actually does make sense, because the data set that I loaded into the system was from endothelial mesenchymal transition. So the endothelial cells lose their normal biological function, and that is shown here because the downregulated nodes are nodes that would be important for leukocyte transendothelial migration, tight junction and so on. But this is actually only one thing here that is downregulated that shows up, so just one hit. This is Claudin 5, which is a tight junction molecule. Actually, so when we click on that, we see that the same node becomes bigger, and when we go to cell adhesion molecules, it will also show up here, because the tight junction molecule Claudin 5 is of course also belonging to the cell adhesion molecules. So then let's go to the reactome. So we reset the view. We go to reactome, downregulated nodes, submit. And there we see actually again, because basically our list doesn't contain so many downregulated nodes, so it's usually then only one or two hits that show up here. But we see here some transport proteins, tight junction interactions. Again, we would see the Claudin 5. And now we go for biological process, calcium independent cell adhesion and so on. So actually you have to look at the p-value. If this is not extremely low, then of course you have to take this result cautiously. So just for completion, I click through these other possibilities here. So if you remember, for the upregulated nodes, it was mainly cytosol and cell surface. And here we see for the downregulated nodes, we see, for instance, transcription factor complex or actin, cytoskeleton, and so on, as more significantly changed genes of cellular compartments. And here we see some microRNAs and some transcription factors when we investigate the motif database. So another possibility that we have here is that we can actually go to Path Explorer. So in this case, for instance, we would like to know what is the shortest path from IFID 3, which is find up here, to interleukin 7 receptor, and that will show you actually that this is the shortest path. And here you see then the how that actually it's one protein that is here linking these two other entities. And finally, with batch selection, you could enter a list of nodes and then select several of genes of interest that you would like to analyze further.